Hi, it's Mr. Andy here, along with my good friend and colleague,
Mr. Brown.
and we're here to talk about the cell cycle and how it relates to cancer. The cell cycle is the process which every cell goes through in its life and is made up of a complex set of stages that are highly regulated by factors both inside and outside the cell. The cell cycle involves interphase and cell division. The cell spends about 90% of its time in interphase, as we can see on this chart here, and the other 10% in cell division, usually mitosis, which is dividing to replace dead cells or repair something in the body. Interphase and mitosis both contain multiple stages each and are regulated by the body.
There are three checkpoints during the cell cycle, with the G1 and G2 during interphase, and the M being during the mitotic cycle or mitosis. Interphase is made up of three different parts. The first part is the G1 phase, which is right here, G1. And this is just talking about, it's mainly the growth of the cell. And at the end of this phase, there's the G1 checkpoint, represented by this red star right here, where the body checks to make sure the cell has grown correctly and completed all the processes it has need to. If the cell grew correctly and fully, it moves on. And if it did not grow correctly or completely, the cell goes to the G0 stage. This is right here, G0. Cells in the and the cell does not divide. Cells in this G0 phase can come back into the cell if needed. And a case where this happened is rebuilding muscles, such as if a muscle was torn, then the body would send the cells back. PDGF, which is a growth factor in the body, and which we will come back to later, attaches to these tissue cells on the outside of the cell, signaling the cell to pass through the G1 checkpoint. Additionally, if this PDGF is not there, the cell will not grow, as this growth factor is an essential part of the nutrients the cell needs to grow. The cell needs to grow during the G1 phase, nor will the cell be signaled to carry on, showing that the PDGF isn't the external factor that still affects the stop and go checkpoint inside the cell. So after the G1 checkpoint, it goes to the S phase. And the S phase is where the DNA, or the genetic material of the cell, is duplicated. And then after this, it goes on to the G2 stage. So this is G2. And here is where the cell just grows again. And after it, you have another checkpoint, the G2 checkpoint. And if the cell was correct and complete when growing, it moves on to mitosis, which is this process down here. However, if it was not correct nor complete, it will go back to this G0 phase. MPF, which is responsible for triggering the passage of this cell through this checkpoint, or MPF is responsible. The M checkpoint in mitosis, which is represented by this star right here, occurs between the metaphase part of it. So I'm just going to put meta right here, meta, and the anaphase part. So this is ana. And the M, well, the body will not proceed with splitting the chromosomes apart and starting this anaphase right here if all the chromosomes, or any one of them, are not connected to the spindle, and the chromosomes are already lined up in the middle of the cell because that happened in metaphase. And the spindle is produced by the MPF, which will be elaborated on later. And if that doesn't attach to them, then anaphase won't start. Or if the MPF does not produce enough spindle, then anaphase won't start either. And what this really shows is that the MPF inside the cell is an internal factor that will affect the stop and go checkpoint in between metaphase and anaphase, or the M checkpoint right here. So this MPF stated earlier in the G2 and M checkpoints is a CDK, and that stands for the cyclin-dependent kinases. This means that it is an enzyme that activates or inactivates proteins in the cell, which is a kinases, and it needs to be attached or bonded to a specific protein in the cell, which is the cyclin, in order to work properly. So this graph shows MPF concentration in the cell. So this is the cyclin production in the cell. And that's not exactly how you spell cyclin. and this is the CDK concentration. So what you see is even though the CDK concentration is constant, the MPF or the concentration or the CDK, like the whole concentration of it, is dependent on the cyclin because this CDK depends on the cyclin. It cannot work without the cyclin. And after the anaphase part, the MPF, which is talked about right here, which needs the cyclin, it helps switch itself off actually by breaking down its own cyclin that's attached to the CDK part, leaving this part useless unless it bonds with another cyclin. And this process allows for the CDK part of the MPF to be recycled so it can be used throughout another cell cycle.
Okie dokie, now let's go and let's talk about cancer. For the most part, when these cells are doing their cell division stages, they work relatively well and they result in extra cells being produced. But sometimes when the cell cycle goes awry and the cell does not go as planned, then we're going to result in cancer, which is going to be an unregulated growth of cells that don't provide their proper function, which will eventually cause death due to interference with cells that are performing their proper function. So these cancerous cells are going to be produced on a fairly regular basis and will most always fail to pass their checkpoints due to their not meeting the required standards and will go off on your G0 phase, which is going to be over here. However, sometimes the cancer cells can make it through the checkpoints just for whatever reason. And those cells are going to be the ones that cause the cancer that we see in everyday life that are going to cause problems. These cells do not degrade like normal cells, and so they can divide and multiply indefinitely given enough nutrients, which causes problems given that they also divide at an expedited rate. These cells also do not respond to the body's control, meaning that they do not have to stay tethered in one place. They may not follow regular checkpoints in mitosis, and they also may not need growth factors to grow and divide. These cancer cells will eventually cause tumors, as we can see in this very unlucky lady right here who has breast cancer. These tumors will start to form, and they're going to be a collection of cancer cells in a given location. They can either be benign, which means they're not going to move from one spot, they're going to stay just in one location, or they're going to become malignant, which means they can move from more than one location, as shown here. And when they start to move to more than one location, that's going to be known as metastasizing. And that means that some of these cells are going to make it into the bloodstream, and they're going to go throughout the body and potentially make tumors in different places, which is going to lead to more problems because you're going to get cancer in different places, such as other than in your breast, you could have it in your pancreas, in your heart, in your liver, which are all going to be things that once you get cancer there, it's not going to be good. And that's...
And we hope that helped.